Hello, everybody. Let's continue the digestive system, a part of the accessory organs of the digestive system. This is Dr. Nadia Fouladian, and we're going to talk about the liver, pancreas, and gallbladder. Those are the accessory organs of the digestive system. Liver, gallbladder, and pancreas. As we discussed before, we're not talking about the mouth, tongue, and tongue, teeth. We're not talking about the tongue, teeth, and salivary glands. That those are also part of the accessory organs, but we're not covering them. So as you see here, regarding the accessory organs, we have the salivary gland. We're not covering that. We are going to talk about the liver, gallbladder that is located behind the liver, as you see here, and pancreas. That's the organ that is located mostly behind of the stomach here. It starts from this loop of the duodenum here and it continues all the way toward the left side, toward the spleen here. Now let's start with the liver. Liver is the largest visceral organ of the body. So after the skin, that is the largest organ of the body with all the vast area of the body that is covered and the heavy skin that we have all around, that's the largest organ of the body. Liver is the largest visceral organ, the organ that we have inside of the body. Liver has many functions that includes the metabolic regulation, hematological regulation, and bile productions. As we're going to discuss these ones regarding the functions of the liver, you will see that liver is acting like a factory for the body, is synthesizing many materials for the body, including many proteins, many polysaccharides, and many other things, right? And with all those metabolic and hematological function and the bile production and many much more function that it has, like the detoxifying of material and all those ones, that's a very vital and essential organ for the body. Now let's go and talk about it more. So first, regarding the metabolic regulation. As we discussed, all the blood that comes from the digestive tract, from the insides of the abdomen, from the small intestine, large intestine, and all those ones, they get into the hepatic portal system and they get into the liver. Now, we have cells of the liver that we call them hepatocytes. Those hepatocytes are adjusting the circulating metabolite before the blood enters into the systemic circulation. So, in other words, the liver is located as a gatekeeper or like a custom that doesn't let anybody get into the whole system of the body. When these materials are on their own way toward the heart to get to the inferior vena cava, and after that get to the right atrium, and then get to the left ventricle, go to the pulmonary circulation, and finally get to the left side of the heart and get spread to all over the body. Before that, this liver here is acting as a surveillance, is controlling them so that the extra material, toxic material, unnecessary material cannot get to the whole systemic circulation. It might become very difficult to spread them from all over the body. So it's things that you just put them at the beginning, catch them at the very beginning and put them away, right? And also the function of that as a storage of the nutrient in the liver. For example, you go to a party, there are lots of delicious foods and you eat a lot, right? Or even for everyday uses, anytime, even you do not eat too much. In every meal that you have, you put lots of material into the small intestine, absorbs, get to the circulation, and now it's going to get to the heart. It goes into the liver, like lots of glucose inside of that, lots of amino acid inside of that, lots of fatty acid, glycerol, and all those things, right? Now, first of all, we do not need to use all of them at once at the right moment, right? So the body has to store them, makes some other material out of that, like for example, liver is synthesizing lots of proteins already, and store them, like from the glucose. You have a surge of the huge amount of the glucose had gone into the blood. Besides the fact that you do not want to use all the insulin for all those glucose that they want to get into the bloodstream at once, you don't want to spend all your insulin of the body, right? And you do not need to use that much of glucose at once, right? So what the liver is doing is just compacting them, combining them, and metabolize that to make the bigger molecules, like for example, polysaccharides, that we call it the glycogen for the glucose, right? Then it stores those ones at the time, like 2 hours, 3 hours, 4 hours later, that do not have access to another source of the glucose at that right moment. Then the liver starts breaking down that glycogen, then sends the glucose into the blood, and at every moment the body can keep a steady level of the glucose so that it doesn't go high suddenly and go very low suddenly, because both of them are dangerous for the body. And it applies to many other things, right? So that's one thing, right? The other thing is that liver has some hematological regulation, is the largest blood reservoir of the body. We are going to talk that when the, for example, hepatic portal system, hepatic portal vein gets entered into the liver, they start branching. They become two more smaller branches, more branches, and they go all over the liver. And there are lots of blood that are coming into the liver. So that's the largest blood reservoir of the body regarding the organ that it has, right? As blood passes through the liver, phagocytic cells remove old or damaged erythrocytes. So that's one function of that we have inside of the blood, phagocytic cells that remove the damaged erythrocytes that sorry. We remember that the erythrocytes sorry. Erythrocytes have a specific lifespan, 120 days, because they are not able to divide and make more, right? So after that, they have to get out of the blood, right? One place that takes them out of the blood is liver, and then in the process that we discussed before, how we recycle the erythrocytes, and they are using different parts of that to synthesizing more material, right? Also, liver cells synthesize lots of plasma proteins, including the proteins that are involved in the transport, such as the albumin, globulin, or the blood clotting factors, for example, or those ones that are helping in the forming of the blood clot formation, right? Those are all proteins that are synthesized in the liver. If we have the liver damage or liver failure, we are losing these functions of the liver too, besides all the other functions, right? So this is the second function. And also the third one is bile production. Pay attention that we said that we have the cells inside of the liver, we call them hepatocytes, sites of the liver. Hepato means liver, site means cells, cells of the liver, right? And bile is produced in these liver cells, in these hepatocytes, one of the functions of all those hepatocytes. So as you see, hepatocytes, or these cells that we have inside of the liver, each one acting as a factory by themselves. They have numerous functions, each one of them. And one of them is synthesizing of bile. Then bile that has produced in the liver comes out of the liver, as I showed you before, that comes through the right hepatic duct and left hepatic duct, and some of that bile is stored in the gallbladder. And then later, when the food comes into the duodenum, bile is secreted into the duodenum when is needed, when the food is gone, especially when a fatty food comes, because we need bile for the digestions of the fat and fat. The function of the bile over the fat and fatty food is that it emulsifies fat. What is emulsification? Emulsification is when you have a big drop of a fat, this bile comes and cover over that one and makes to get break down to the smaller pieces. That's the one that we call it emulsifications. And it happens in the small intestine when you have the fat into the lumen of the small intestine. This emulsification process makes it easier for lipase that is coming, for example, from the pancreatic lipase, to go and act over the smaller pieces of the fat and do the actual digestion of the fat. Break down the fat like the triglyceride we break down to the fatty acid and glycerol, right? So it makes it easier for the digestion of the fat. And so that's why we need the fat. We need the bile. We need fat too, but we need the bile to digest the fat. But to understanding the function of the bile, but after that that bile is emulsifying, then also is covering those parts of the fatty acid and glycerol too, so that they can be transferred into the small intestine too. And those are the times that we call them micelle. So these bile, because the fat is not water soluble, so that's not easy to pass through the cells, right? So that one, for the transportation actually of the fat and all the ingredients of that, these biles comes and covers around that. The bile is acting like a soap, right? You have like when you have a drop of fat over your clothes, for example, you want to wash it. If you just wash it with water, it cannot be dissolved in the water to go away. What do you use? You use a detergent or the soap. And the soap is the one that makes that drop of the fat soluble in the soap, and like that one can be washed away from that cloth, right? So that's actually the way that the bile is formed. The specific thing about the soap is that it has the molecules that we have over there. They have one end that is water soluble, one end that is fat soluble. The fat soluble ones that both get close to the fat drop, the water soluble ones get close to the water one, right? And in this way they can pull the fat out of that cloth, right? So that's the one that is binding. This has one end that can go get close to the drop of the fat, and it has another end that can go be around the water. And in this way, it can transfer the fat inside of the water. That's why we need bile for the breakdown and also for the better transportation into the small intestine. So now, regarding the liver, for the anatomy of the liver, gross anatomy of the liver, we have 4 lobes, right lobe and left lobe, that you can see that from the anterior view. And we have 2 other lobes, caudate lobe and quadrate lobe, that they can be seen when you look from the inferior, or you push it up, right? We have one anterior surface and one posterior surface for the liver. Now, as we discussed before regarding the double layer of the peritoneum that comes between the diaphragm and goes toward the anterior surface of the liver, we said that we call that falciform ligament. that marks the boundary between the left and right liver. That's the place that this falciform ligament is connecting to the liver. The inferior portion of the falciform ligament becomes thick and covers around the structures that altogether is called the round ligament. That round ligament actually is a remnant of a vein that has used to be in the fetal period, has used to be umbilical vein of the fetus, fetal umbilical vein. And that was the vein that was bringing all the material from the mother that was passing through the umbilicus to get into the liver, right? Liver of the fetus, right? So that was regarding of the fetal period. After birth, those umbilical vessels, they all go away. We do not miss them. And the remnant of them between the umbilicus to the liver becomes useless, becomes a ligament because no blood is passing through them anymore. So that was regarding a round ligament. Pay attention that we have another name for that we called it ligamentum teres. Ligamentum teres. And the falciform ligament spreads over the surface of the liver, attaching to the inferior side of the diaphragm, that it spurs little parts of the liver uncovered. And that's the one that we call it coronary ligament. This spreading ligament is called the coronary ligament. Now let's see the picture of that here. This is the falciform ligament. In this area, a double layer that it connects to the anterior to the diaphragm that is located in front of that. Then when it goes up and spreads here, these are called coronary ligaments here. And then on the bottom, it continues over this ligament that is remnant of the umbilical vein and all together makes a round ligament. Now, for the blood supply of the liver, we have two blood vessels that supply the liver, hepatic artery proper and hepatic portal vein. Pay attention that hepatic artery proper comes from the aorta. It separates from the aorta and comes toward the liver. This one brings the oxygen and nutrient from the aorta toward the liver. For the use of the liver as an organ, it needs to have glucose, oxygen, and all those nutrients, right? The hepatic portal vein, as we discussed, when it comes toward the liver, it brings all the nutrient toward the liver, but it's not for the personal use of the cells of the liver. As we discussed, it's for those nutrients to be processed, packed, and all the other function that is done over that one. And then, after the liver, blood gets out of the liver and returns to the inferior vena cava through which vein? Hepatic vein. So make sure you're differentiating between the hepatic portal vein that comes into the liver and hepatic vein that leaves the liver and pours into the inferior vena cava. Right under the diaphragm, on the very top over there, it pours into the inferior vena cava, and inferior vena cava, as soon as it passes through the diaphragm, gets into the heart. To which chamber? To the right atrium. Then blood from the right atrium continue its way inside of the heart. Here you see the hepatic artery proper, that is a branch of the celiac trunk and supplies the liver. Now, after the gross anatomy, let's go over the histology of the liver. If we see the liver under the microscope, what we see? The liver is divided into approximately 100,000 structures. We call each a liver lobules. So these are like building blocks of the liver, right? Each lobule is separated by a septum. We call it interlobular septum. Then in the center of each lobule, we have a vein a vein in the center. What should we call it? Central vein, right? The center of the lobule, central vein. Now we have the cells of the liver, hepatocytes. They are arranged in rows, in cellular lines that they extending from the central vein toward the out, toward the periphery of the liver lobule. So here you're looking at the structures of the lobules. This is one lobule, hepatic lobule or liver lobule. And this is the next one, that is the next one, that is the next one. You see you have some septum. These septa that are between those lobules, because interlobular means between lobules, right? Interlobular septa. Now you see that these lobules are hexagonal. They have 6 sides, right? And at each corner of them, you have 3 structures. That these 3 structures, they are called triad. And what do we call that triad? We call them portal triad. Portal triad is coming from one artery. This artery is actually a branch of the hepatic proper artery. We have one vein that is a branch of the hepatic portal vein. And we have one bile duct that is coming, that is finally making the common bile duct. We are going to talk about all these ones in detail, but this is good to see that these 3 structures are branches of the structures that you saw in the lesser omentum, in the hepatoduodenal ligament of the duodenum. They were coming toward the liver. But just pay attention that artery is coming toward the liver from the aorta, bring the oxygen and nutrient. Vein is coming toward the liver from the hepatic portal and brings all the nutrients from the digestive system, right? But this bile duct is actually a source from the inside cells of the liver, and then it comes out, make this one, and this one goes out of the liver, and finally is making the common bile duct. So pay attention to the different directions of the movement of the material. Now here in the middle of the lobule, before going back, use each one of these structures. They are called interlobular. You have interlobular vein, interlobular bile ducts, and interlobular artery, right? But you know that what is the origin of the artery and vein, and what is the destination for the bile duct. Now, in the center of the lobule, you see the central vein, and you see that the cells, hepatocytes, that you see them as these cuboid cells here, each one is one hepatocyte. They are arranging in the rows or lines, cellular lines, that they start from the center, goes toward the out, like the rays of the cells that go outward, right? Now, between those lines of the cells, you have some spaces that we call them sinusoid, and we have some capillaries, the branches of these arteries and veins that get into these spaces. Those capillaries, those tiny branches that they are capillary, goes into these spaces. We call those capillaries as sinusoid capillaries. Do you remember what type of capillary are sinusoid capillaries? Those are the ones that they have big cleft on the walls of them. We do not have much of basement membrane, and there are big cleft between the epithelial cells around them. Therefore, the material that they are coming, they can easily get into from the capillary, get into the hepatocytes, or after they have been processed inside of the hepatocytes, they can get back into those capillaries very easily, because there are large gaps in the wall of the capillary, so that the bigger material can pass through them, like the protein that have been synthesized, or the glycogen, or the other things, right? So when the material, as these are passing, they make interaction with the cells, send some material, receive some materials, and blood continue its way to get to the central vein, right? Now you have central vein from each lobule, right, in the middle. All these ones at the end are connecting to each other. They become bigger branches at the very end. When they get all together, they make big veins. What do we call those veins? We call them hepatic veins. That's the vein that gets out of the liver and goes into the to the inferior vena cava and finally gets the blood toward the heart so that in the next round of the contraction spreads all the blood that now it is ready, toxic free, and completely have been have been under surveillance and is harmless and useful get all over the body, right? So that's the functions of the liver that is doing to the specific structure that you see. That is totally, again, this is another example that how a structure is serving a function. Now let's just review those ones. Spaces that are created between the lines of the hepatocytes are called sinusoid. Sinusoid consists of sinusoid capillaries that lead to the central vein, and we have some cells that we call the satellite macrophage or Kupffer cells. Those Kupffer cells, the specific name for the macrophages that we have in the liver. Those are phagocytic cells of the liver. They just get rid of the debris and all those ones. Let me get back here. I don't have, I think maybe in the next pictures I have it. So we have those Kupffer cells, right? Now, each lobule of the liver has a hexagonal shape, as you saw. At each of the 6 corners, we have a triode, interlobular vein, branching the hepatic portal vein, interlobular bile duct, interlobular artery, that is branch of the hepatic artery proper, and all 3 branch form the portal triode. That's here. That's a good picture. You see one corner, you have all those 3. You see all the capillaries that are making here. And here you see that in the middle of the capillaries, you have these Kupffer cells or satellite macrophages. Now, this is a good picture to see that each cell, each hepatocyte that you have here, besides all the other function, is producing bile. These green ones that you see here, they are bile that have been produced. And here have been shown that how these biles are coming out of each cell, and finally they go to the small canals that we call them bile canaliculi. Those canaliculi are connecting to each other, and finally they are going into the interlobular bile duct, and they continue their way. Again, they are connecting to each other, become bigger. Finally, they get out of the liver as the right hepatic bile duct, and left hepatic bile duct. Those are the final parts of the bile canals and canaliculi that they are coming out of the liver, and we talked about the rest of that, how they making common hepatic duct and they connecting the cystic duct to make the common bile. Okay? Now, this is just we discussed that hepatocytes are producing bile, so just pay attention that production of the bile happens in the liver, but not in the gallbladder. Bile enters the bile canaliculi that travels to the bile ductus, those biles that the small ducts that I showed you, which connect to an interlobular bile duct in the nearest portal triode. So just emphasizing here, these are ductal, here this one. And here, sorry. And then bile travels to the left and right hepatic ducts and travels to the common hepatic ducts to the duodenum. or travel through the cystic duct into the gallbladder for storage. So regarding this one, when the bile finally gets to this hepatic duct and the common hepatic duct, continue its way and goes toward the duodenum. It remains behind this sphincter until it gets open and the bile pours into the chyme that comes into the duodenum, right? Then, when the chyme passes through here and goes to the waist and the duodenum is empty, this sphincter gets closed because there is no need for the bile to come out, right? When this gets closed, the bile gets back up here, and as goes here, part of that gets into the gallbladder. And gallbladder is a space for the storing of the bile here. So as you see, it gives an extra space here for the bile. Instead of the bile to get back and just reflect back inside of all the ducts inside of the liver. We do not like that one happens, put pressure over the cells of the liver hepatocytes. Instead of that, it just goes into the gallbladder and stores over there for the next one. Besides storing over there, gallbladder dehydrates the bile. It makes that concentrate. When the bile gets concentrated, it just occupies less space so that we can have and accommodate and stores more bile in the gallbladder. Again, in the next one, when the chyme comes here, among all the signals that it sends through the cholecystokinin to go and open the sphincter here and the bile comes here, cholecystokinin also acts over the wall of the gallbladder here and makes that to be squeezed. And the contraction over the wall of the gallbladder, it makes the bile, the concentrated bile now, to get over the cystic duct and gets down and get into the duodenum. So you see that how the cholecystokinin is acting over the biliary system to make it to enter to the duodenum after the food has entered to the duodenum. So that was regarding the liver. We talked about the liver. Now let's talk a little bit about the gallbladder. Gallbladder is divided into 3 regions: fundus, body, and neck. And as we said, this cystic duct leads from the neck of the gallbladder to the common hepatic, connects to the common hepatic duct and makes these two together make the common bile duct, as you saw here. So here you have the fundus here, this part, this final part. Then you have the body here, and then you have the neck here. Now neck is connected by the cystic duct. It connects to the common hepatic duct, and these two are forming together common bile duct here, as we discussed. As you're looking at the gallbladder here, pay attention to the shape of the mucosa of the gallbladder. So gallbladder function is important to understand that function of the gallbladder is a storing of the bile, storage of the bile and the concentration of the bile. But as we discussed about that, but it is not production or synthesis of the bile. That's the job of the liver. Now regarding the bile storage and modification, when the sphincter of ampulla, that is the hepatopancreatic ampulla, or the other name for that is sphincter of Oddi, when that sphincter is closed, then bile enters the cystic duct and into the gallbladder, right? The gallbladder can store about 40 to 70 ml of the bile, and then water is taken away from that, is continuously removed from the stored bile, therefore concentrating the bile more and more. Now when the food, or the chyme as we call it, entering the small intestine, when especially if we have the higher fat content in that, the small intestine cells, will release cholecystokinin. And the cholecystokinin, you remember we said that the cholecyst is another name for the gallbladder. And we abbreviate that as cholecystokinin as CCK, cholecystokinin. That one will cause the gallbladder to release the bile. As we discussed, it just goes and acts over the wall of the gallbladder to contract. It will cause the gallbladder to contract to release the bile. Also, it causes the sphincter of ampulla to open. And all these two helping the bile to get into the duodenum. Bile enters the cystic duct from the gallbladder, enters the common hepatic duct from the liver, and the sphincter of ampulla opens. Bile enters the duodenum of the small intestine and then will emulsify the fat as we discussed it. And here again it shows that how the bile that is coming here, when the sphincter is open, it just gets into the ampulla and through the papilla gets into the duodenum. Now, regarding the histology of the gallbladder, the wall of the gallbladder consists of the mucosa, lamina propria of the mucosa, muscularis layer, and serosa only. So as you see, we do not have all the layers that we have in the wall of the other parts of the digestive system, because gallbladder simply has much thinner wall than the other parts, right? There is no muscularis mucosa and there is no submucosa here. So pay attention to that one. And mucosa has folds that separate the mucosal glands. Those are the ones that you saw inside of the mucosa, those folds that are making those specific shape inside of the mucosa. A muscular layer has both circular and longitudinal smooth muscle layers. Now, that was regarding the gallbladder and its function and structure, regarding the gross anatomy and histology of them. Now let's have a few words regarding the pancreas as another accessory organ of the digestive system. Pancreas is located posterior to the stomach, most of them. And the pancreas consists of one head that is nearest the curvature of the duodenum. In this C curve of the duodenum, the head of the pancreas located exactly in that one. And that's the best way to find out the head of the pancreas. Search for the duodenum, the head of the pancreas is located there. Otherwise, sometimes might be difficult to find it. Then it has a body, the main part that extends toward the spleen, and the tail is the part that is close to the spleen, rounded end of the, or the tapered end of the pancreas that is near the spleen. We have a duct in the middle of the pancreas, pancreatic duct, that delivers exocrine secretions of the pancreas to the duodenum. As you remember, pancreas is like double organ. One endocrine part that makes that part of the endocrine system, and one exocrine part that is making that part of the digestive system. We talked about the endocrine part when we were talking in the endocrine system. Now here we're talking mostly about the exocrine part. So those exocrine secretions, you remember that exocrine glands are the ones that they have some secretion that pour into a duct, and that duct leads into a lumen somewhere, right? And here we have a duct as all those tiny ducts that come from the different parts of the pancreas, different exocrine parts of the pancreas make a main duct that we call it pancreatic duct, right? And those pancreatic duct finally enters into the lumen of the duodenum. And sometimes, besides that main pancreatic duct that we have an accessory pancreatic duct that I will show you in the next pictures. Now, arterial blood that enters through the pancreatic and pancreaticoduodenal arteries and drains through the splenic vein. So blood vessels, arteries are pancreatic artery and pancreaticoduodenal artery, and the vein for the pancreas is the splenic vein. As you remember, splenic vein is part of the hepatic portal system that joins all the other two superior mesenteric and inferior mesenteric vein and makes the hepatic portal vein. So here you're looking at the blood vessels that we have here, that you see that you have these pancreaticoduodenal arteries supplying the pancreas. Here you have superior pancreatic artery that is supplying that one. And also you see that these other vessels, like for example, even some branches of splenic artery are coming and supplying the pancreas. Now, regarding the histology of the pancreas is composed of some lobules. And inside each lobule, we have some exocrine part that we call them pancreatic acini, and some endocrine parts that we call them pancreatic islets, right? For the acini is a plural for the acinus. So these are the ones that are producing digestive enzymes and ions. And these all together are called pancreatic juices. So these are the secretions of the exocrine parts, that those are acini. These enzymes travel through the pancreatic duct to the small intestine, right? Pay attention that pancreas also is secreting some bicarbonate that acts as a buffer, and that's among all the secretion that enter into the duodenum to make that basic environment and take the acidity away. And those are all parts of these pancreatic acid. Now, pancreatic islets that are among the acini, pay attention that most of the cells that we see in the pancreas, they are acini. And regarding the size, pancreatic islets, the whole size of the islet are much less than the surface that is covered by the acini. So these islets, those are the ones that are producing hormones, and those are the endocrine parts. Hormones enter into the bloodstream to travel to the target organ. Whenever they reach the target organ, they start acting the designated function that they have, right? So here you're looking at the drawing of the part of the pancreas. You see that you have all these acini here. Each one is called an acinus, all acini. Then each one has a duct here, that these are all finally connect to a bigger duct that make the main pancreatic duct here. And among all those ones, you have these islets, that those are the endocrine parts, and those endocrine parts have 4 types of the cell, as we discussed in the endocrine system, alpha, beta, delta, and F cells here, right? Remember? Now here you see the actual histology under the microscope, and then you look here, you see that most of the tissue of the pancreas, they are all acini, that they are exocrine part, all of these ones, right? A few one have been circled so that grab your attention, but they are all the same, right? And among them, you have some islets, those are the ones that are endocrine parts. And you see the sections of some of those ducts here. Now, regarding the exocrine secretions of the pancreas, that they are digestive enzymes and some buffer, you have all these enzymes, pancreatic enzymes that come from the acinar cells. These enzymes actually can break any type of the big molecules. Like we have, you remember when we have ACE at the end of the name, means that is able to break down, that is an enzyme that is able to break down that material, right? Like lipase is the enzyme for breakdown of the lipid, digest the lipid. We have in the pancreas, we have all these ones. We have lipase, we have carbohydrates to digest the carbohydrate, we have nuclease to digest nucleic acid, and we have proteolytic enzyme to digest the protein. That these ones include proteinase and peptidase, means that they are able to break the proteins, the complete protein, or they are able to break down the peptides. Peptides are the small branches that they finally making the proteins. The collection of the amino acids together, when they attach to each other, they are called peptide, right? So as you see, the pancreas has all types of the digestive enzymes. So just a few words regarding the pancreatic hormones, that they come from the pancreatic islets. You already know about these ones, that the insulin is a hormone that acts over the glucose, for example. Glucagon, we talked about that is a stress hormone that brings the level of the insulin, brings the level of the glucose up inside of the blood. Insulin brings the level of the glucose down by helping them to get into the cells. And somatostatin and for example P-peptide that we discussed before. So for the regulation of the pancreatic secretions, cholecystokinin, as we discussed, from the small intestine will cause the pancreas to release its digestive enzymes. We talked about all these ones, right? So we already talked about how cholecystokinin acts over the bile to help the bile to get into the duodenum. Now here you see that the cholecystokinin also acts over the pancreas to release its digestive enzymes through the duct, pancreatic duct, toward the lumen of the duodenum. On the other hand, so this one acts over the digestive enzymes, and secretin from the small intestine will cause the pancreas to release buffer that coming from the pancreas. When the food leaves the stomach and enters the duodenum, the chyme is mixed with the acid in the stomach, as we discussed before. Therefore, acidic chyme is entering the duodenum. Buffer are used to maintain a normal small intestine pH that is about 7 to 8. As you remember, the pH environment of the stomach, it was acidic, and we see that the duodenum, small intestine, is not acidic anymore because of those buffer that come from here and also from the duodenum wall itself, as we discussed before. So the release of the buffer is under control of the secretin. The release of the digestive enzymes are under control of the cholecystokinin. So that was the end of the whole chapter of the digestive enzymes, and hopefully you enjoy it and learned all of them. So we will see you for the next chapters. Have a great time.